Think Forward. Think Research Channel.
I'm David Henderson. I'm the Deputy Director of our hospital, and I'd like to welcome you all to the 2002 Medicine for the Public Lecture Series. As I said, this is our 26th year of these programs, offering these programs for the citizenry of Bethesda and the surrounding environment. These lectures are really designed to bring you in to what we do, to give you a chance to see what's going on our campus. I came here, I hate to admit it, 20-some years ago, like a lot of people who come here thinking I would stay for 2 or 3 years and then go off and get a real job, but just got hooked by the place. And I hope you have the same experience tonight as we hear about communication disorders. So now let me tell you a little bit about tonight's speaker. Dr. Drayna completed his undergraduate training in biology at the University of Wisconsin in Madison. He then went on to a small school in New England called Harvard, where he received his PhD in microbiology and molecular genetics in 1981. From 1981 through 1985, he was a postdoctoral fellow at the University of Utah Medical Center through the Howard Hughes Medical Institute in Utah. He then worked in the private sector for some biotechnology firms that you've probably heard of, such as Genentech. He holds 5 patents related to gene products and genetic diagnostic tests. And he joined us here at NIH in 1996 as a first visiting investigator with the National Center for Human Genome Research, and then later joined the National Institute on Deafness and Other Communication Disorders as a senior fellow in 1997. Currently, he's a senior fellow with the section on transcription factors, sensory receptors, and channels, the Laboratory of Molecular Biology in the NIDCD. His research interests focus on using genetics to help us understand the factors causing inherited speech and hearing disorders. And tonight he's going to talk to us about the genetics of speech and communication disorders. Please join me in welcoming Dr. Drayna. Dr. Drayna.
Well, thank you very much for that very kind introduction, and I would like to add my welcome to you all here to the NIH this evening with my compliments on those of you who were able to persevere through the security inspections that we now all deal with every day. So it's very nice to see you all here. And I'm looking forward to telling you about not only exciting work here at the NIH, but more generally in the field of the genetics of speech and communication disorders. So I'm going to start simply by defining these. What are they? Well, these are generically disorders of voice, speech, and language. These disorders often arise in childhood where they're common. All right? By common, I can tell you, for instance, one popular statistic is that as many as 20% of young children have recognizable speech and language disorders at some point in young childhood. Now, many of these can improve with therapy or spontaneously, although in a fraction of individuals, they persist through adulthood. And in fact, the numbers, given the commonness of some of these disorders, the numbers that ultimately persist through adulthood are quite substantial. One of the things I'll be speaking tonight about is stuttering. It's estimated that there are 1 million individuals in the United States who stutter as adults. So these are very significant in terms of the number of individuals they affect. What causes these disorders? Well, I've really divided this into 2 general areas, the so-called non-specific causes, and I've put a couple of them up here. For instance, trauma can cause certain speech or language disorders, although this is relatively rare. Things like low birth weight, perinatal adverse events, very generalized, non-specific problems, insults to the central nervous system that can, in some cases, lead to speech and language disorders. Another good example of a non-specific cause is hearing loss. It's, for instance, a well-known fact that if a child is born with no hearing, if they don't hear, they don't learn how to speak. All right? So that, in a sense, that communication disorder is secondary to their hearing loss, but it represents a significant fraction of all communication disorders in humans. And then there are the specific causes, and it's clear there are some very specific, precise causes of speech and language disorders in some cases. However, the ultimate underlying nature of these causes is currently not known. All right, so what might some of these other factors be? Well, I'm going to talk to you tonight about one of the more prominent other factors, which is inheritance, or so-called genetic factors in these disorders. So everyone here, I see we have a lot of young people, everyone here will hopefully, in addition to hearing a little bit about speech and language disorders, come away with a basic understanding of human genetics. Genetics in general and human genetics in particular, and how we use it to understand biological phenomena. Okay, what is genetics? Well, I think the best definition of genetics is this, which was actually sort of produced by Victor McCusick, a very distinguished, recently retired university professor at Johns Hopkins. It's the study of variation. It's differences. We are all very much alike, and at the same time, we are all unique and distinct. And much of that uniqueness and distinctiveness comes from what we inherit. There are a large number of genes that we all carry around in our chromosomes, and there is a very large amount of normal variation that exists in those genes. And that variation is passed down, it's inherited, according to simple rules, all right? And this variation can be used as the source of basic understanding of all sorts of biological functions. And I hope to leave you this evening with an understanding of how naturally occurring variation in humans can be used to understand important biological functions that we have, okay? All right, so how do we do this? How do we use genetics to learn about nature? Well, I have summarized a very large and complex undertaking in 4 bullet points here for your convenience this evening. Basically, there are 4 essential steps to this process. The first is to identify variation that is passed down from parent to offspring. There's a lot of variation among all of us. Not all of it is passed down from our parents. Some of it comes from other causes. Things like environment, random factors, sometimes called stochastic factors, can have a significant effect on this variation. But in order to do this, to use genetics as a tool to understand things, we have to find variation that's passed down, okay? Meaning it's genetic. We have to show this variation is due to genes and not environment or other factors. We then identify the genes which, in variant form, cause the visible trait. And here's the first lesson that I hope everybody's going to leave with tonight. I'm going to leave you with one genetic term, one take-home term. The visible trait is what geneticists call the phenotype. All right, and you might hear me use this phrase as we go through this evening. The phenotype is the part of us that you can see, that you can measure. And it's distinguished from the genotype, which is the actual genetic variation itself, that in fact is not easily visible. All right? All right. Finally, when we find these genes, the real reason, the genes by themselves are in fact of limited use. The real reason we want these genes is to study what they make. What are the product of these genes? To learn what molecules, structures, and functions are involved. Okay? So that's the general procedure that we as human geneticists undertake. And I'm going to spend some time actually covering all of these now in succession. I'm first going to talk for about a third of my talk about genetics in general, all right, to get everybody here on the same page with what genetics is and how we use it to do this. All right. So what's the first problem? Well, the first problem is what's genetic, all right? There are lots of things that cluster in families but are not due to genes. A good example is being Episcopalian. Clusters very strongly in families, but is probably not due to what you inherit, all right? How do we know that speech and language disorders are due to genes and not other causes, all right? So the genetic contribution to a trait is measured in several ways, all right? And I'm going to describe these because, in fact, I'm going to go into all of these in more detail later in my talk. The first way that we measure the genetic contribution is what's called a twin study. It's a classical method. It's been around for, the better part of 100 years. And a twin study does the following. A twin study compares the occurrence of a trait in identical twins who share all their genes to the occurrence of that trait in fraternal twins which share half their genes. Okay? So you can imagine if a trait is 100% genetic, all right, then identical twins will always be alike. All right? So if, for instance, hemophilia, which is a genetic disease, a genetic disorder, if one member of an identical pair of twins has hemophilia, the other one always has hemophilia, 100% of the time. All right? Other disorders are not necessarily like that, and when you see a disorder that is not always the same in identical twins, that suggests other things must be involved, other things other than genes, because these identical twins are in fact clones of each other. They are genetically identical. All right? So, twin studies are very powerful. There are several different types of twin studies. There are a number of different designs. I'll give you an example of a very powerful design. Identical twins reared apart. All right? Well, identical twins reared apart share, in general, no environment. They have no environment in common. There are cases of identical twins that were raised 2,000 miles apart and really had nothing in common in their environment when they were growing up or as they live as adults. And so, anything that is the same in those people is unlikely to be due to environment because their environment is not the same. Right? It's much more likely to be due to their genes because their genes are the same. All right? The problem with that particular study design is that identical twins reared apart are very rare. There just aren't many of them. But more on that later. What twin studies can do is they can estimate how much of a trait is due to shared genes versus how much is due to other factors, such as environment. All right? Now, what's the second way we know or measure how genetic something is? Well, the adoption study is another very powerful tool. In an adoption study, we compare the occurrence of a trait in adopted children to its occurrence in their biological parents, to whom they are genetically related, and in their adoptive parents, to whom they are not related. Now, in the United States, adoption records are closed. All right? So you can't very easily put together adopted children with their biological parents. In Scandinavia, for instance, adoption records are open, and in fact, it's quite straightforward to do this, and a lot of adoption studies are in fact, have been pursued very successfully in places like Scandinavia. In a place like the United States, really all you can compare is adopted children to their adoptive parents, which sort of gives you half of the equation, but sometimes that can be quite informative, all right? And again, I'll give you examples of this later. And then finally, I'm going to talk about a segregation study. A segregation study is a very specialized tool in human genetic analysis. And in a segregation study or a segregation analysis, you compare the occurrence of a trait in a family to what you would predict for a purely genetic trait. All right? So traits that are due to our genes are handed down, passed down from parent to offspring according to a set of simple rules. These rules were initially figured out by a fellow by the name of Gregor Mendel, working in garden pea plants in the 1860s. But it has subsequently been shown that these rules apply to all higher organisms, including humans. And for instance, being Episcopalian, well, it's been shown that religion clusters very strongly in families. But if the parents are Episcopalian, it's very likely that all of the children in that family will be Episcopalian. So in other words, religion doesn't cluster like a genetic trait. Everyone in the family tends to be the same way, whereas in a genetic trait, some people are affected and some people are not, according to very specific rules, all right? And a segregation study looks at this very carefully over a large series of families and compares the occurrence of a trait and can help you decide whether it looks genetic or not, all right? So, how genetic are speech and communication disorders? So most of these methods, in fact all of these methods, have been applied to speech and communication disorders. I, for one, work on the genetics of stuttering, and so I'm going to talk a little bit about stuttering as an example here. All right, so for instance, stuttering, a number of twin studies have been done of stuttering. And there's a lot of variation from study to study because they're done in different countries. There are different definitions of what stuttering is in different countries and different cultures. But in general, the studies paint a fairly consistent picture worldwide. And that is, identical twins, stuttering occurs in both identical twins at a rate about 2 to 3 times higher than both fraternal twins of a twin pair, all right? So the occurrence of something, when identical twins are more alike than fraternal twins, that's an evidence that genes are involved, all right? Adoption studies, well, adoption studies have mostly been done in the United States, so they're of the one-sided variety that I mentioned earlier. And the one clear conclusion from adoption studies is that the occurrence of stuttering in adopted children raised by stuttering parents is no higher than normal. All right? Now, these stuttering parents are not related, genetically related to these children. All right? So what that tells us is that children don't learn how to stutter by listening to parents who stutter. There's really no evidence that people learn how to stutter. All right? And an adoption study is a fairly powerful way of asking that question. All right. Third thing is familial clustering, which is a type of simple segregation analysis, all right? And about half of individuals, in fact almost exactly half, of individuals who present themselves for stuttering therapy report a first-degree relative who stutters. Now by first-degree relative, I mean either a parent, a sibling, brother or sister, or an offspring. Well, that is much, much higher than the rate reported by the general population. It's really just a few percent of the general population that has a first-degree relative who stutters. But if you stutter, there's a 50% chance that one of your first-degree relatives who stutter. That's a many, many-fold elevation of risk, as the statisticians say, and argues very strongly for a genetic component to stuttering, or at least some of stuttering. All right, and finally, genuine, pure segregation studies for stuttering have been done, and I'll just tell you, they have not been conclusive. They are mixed. Some studies support the idea of a clear genetic mode of inheritance for stuttering, especially in a few special families that I'll talk about in a minute. But many segregation analyses of stuttering have really not produced conclusive results. Okay? So the take-home message here is that there's very clear evidence for a genetic contribution to stuttering, or at least some of stuttering. But exactly what that contribution is not so clear at this point. All right. I want to talk a little bit more about other communication disorders. Now, the state of this field is such that many of these disorders are not particularly well characterized. All right? They often lump these speech disorders together as a single group. And in these studies, this can include all sorts of things which are probably quite different from each other, clinically quite different from each other. But all of these studies suggest that these disorders as a group have strong genetic influences, all right? Of the sorts of types that I mentioned before, twin studies, segregation analysis, etc. Now, in addition, I must mention that there are relatively rare genetic syndromes that also affect speech, plus other things. Okay? Hereditary hearing impairment is a classic example. About 1 in 1,000 children are born deaf in the United States, and most of these, very large amount, is caused by hereditary deafness. And in fact, many of these children will have subsequently, due to their deafness, very profound communication disorders. Okay? Likewise, for instance, there's a disorder called neurofibromatosis, which causes tumors of the nerve system, frequently in the auditory part of the brain, sometimes called acoustic neuromas, that can have strong effects, secondary effects as communication disorders. All right, so I hope I've made this clear to you that why we believe there are genes involved in speech and communication disorders in humans. All right? It may not be as clear as simply inherited disorders. Many of these do not follow simple patterns of inheritance as laid out by Mendel 140 years ago, but it's clear genes are somehow involved. We know that. All right, so if we know that genes are involved, how do we go about finding these genes? How do we do it? All right, and why? Okay, first of all, I need to introduce the other term I'm going to leave you with tonight, which is positional cloning. It's done by positional cloning, is the short answer here. Finding genes is done by a process known as positional cloning. What's that? That is the process by which a gene is identified solely on the basis of its position. That is, what place on what chromosome. The reason for that is because now, thanks to the Human Genome Project, we know all of our genes. We know where they all are on each of our chromosomes, and if we can tell the place at which a gene resides, we can ultimately find that gene, even if we don't know anything else about that gene. All right? Knowledge of what the gene does or how it does it is not required, but this method has allowed the identification of genes that underlie hundreds of different human disorders. And in fact, the great triumphs of human genetics of the past decade were, in many cases, the result of this process, positional cloning. All right? So, how do you do this? What exactly is it? All right. Well, the way we find the location or the position of a gene is we begin with something called a linkage study. And now I'm going to drag everybody here back to introductory biology. What is linkage? All right. Linkage is the co-inheritance of traits as they are passed from parent to offspring. All right? Gregor Mendel, this guy in modern-day Czech Republic, elucidated the laws of inheritance in studies in garden peas in the 1860s. One of his laws was his second law, now called his second law, which is sometimes called, although he didn't use this term, the law of independent assortment. And what that says is the following. Pairs of traits are passed from parents to offspring in random combinations. Okay? So, for instance, some of the things Mendel studied were plant height. Is it a tall plant or a short plant? Another thing Mendel studied was the seed color. Are the seeds green or yellow? Well, if you take, for instance, a tall plant with green seeds and you cross it, you mate it to a short plant with yellow seeds, and you breed the offspring, eventually you get all combinations in the offspring. You get tall with green and tall with yellow and short with green and short with yellow in even combinations. All right? That's Mendel's second law. Every once in a while, however, that fails. Linkage is an exception to that rule. In rare cases, traits are found to always be transmitted together. So in this example, all offspring pea plants would be tall and green, or short and yellow. When that happens, the height gene and the seed color gene are said to be linked. all right? These are rare. Mendel never saw them. That's why he postulated the law of independent assortment, and in most cases, the law of independent assortment is completely true. But in rare cases, linkage is observed. Why? Linkage occurs because the genes that code for the two traits lie immediately next to each other on the same chromosome. These genes lie so close to each other that they are in fact not very susceptible to the normal mixing process that all genes undergo as they are passed from parent to offspring. Okay? Everybody with me here? Okay. Scientists, now, to do linkage studies in humans, we don't use tall humans and short humans. It turns out that's not a very good trait. And we certainly can't use green humans and yellow humans. We, thanks to the Human Genome Project, have available to us an almost infinite supply of genetic markers that are even better. All right? They are at known locations. There are thousands of them. And if a trait shows linkage with a marker, since we know where this marker resides a priori, the gene causing that trait lies nearby. All right, now I'm not going to bore everybody here with what these markers are and exactly how we measure them, but it's done in a molecular biological laboratory using polymerase chain reaction and automated DNA sequencing machines, and if anybody wants to know more, I'd be delighted to explain further after my talk. But just take this one on faith, if you can, that these genetic markers are just as good as Mendel's tall and short and green and yellow. Only they work for humans, and they can be easily assayed at many positions up and down all of the chromosomes. Okay, so how do we do a linkage study? Well, in practice, we gather DNA, usually from blood is the most common method, and clinical information from family members, that is, who's affected with the trait. It could be a genetic disease, or it could be something else, and who's not affected, right? Then we type these DNA markers in across all the chromosomes in all the individuals. So we use, just for practical purposes, about 400. So we start, for instance, at the top of chromosome 1, and we have a marker, and we look at that marker in a family where a disease or a disorder is being transmitted, and we ask, is this marker co-inherited with the disorder in this family? And true to Mendel's second law, the answer is almost always, no, it's not co-inherited. It's random. Okay? So we go through hundreds of these, and somewhere along the line, if a disorder is genetic, we'll find one, in fact, where it is co-inherited. All right? And we then have discovered linkage. Now, since we know where these markers are, we then know approximately where that gene is. Now, I should tell you that since human families aren't like peas, garden peas, or fruit flies, or mice, you don't have complete control over what a family is or looks like. There's usually a lot of missing information. So in fact, we do this, it's required to do a statistical analysis to determine the degree of co-inheritance between the trait and each marker. But a high degree of co-inheritance now equals linkage, right? All right. So we found linkage. We know the neighborhood where this gene is, starting from just the fact that the trait was inherited in families. Now we know where this gene is located, approximately. How do we find the gene itself? Well, again, I've summed up many, many person years of work in 4 easy bullet points. First thing we do is we identify the smallest region of that chromosome that shows complete co-inheritance with the trait. All right, we know the gene must lie in this little piece of DNA on some part of some chromosome. All right. Then we identify all the genes that are in there. We need all the potential bad actors. We need the whole cast of characters. We test every single gene to look for differences in that gene in affected individuals compared to unaffected individuals. And the basic indication that you have found the gene that causes the disorder is that you find DNA sequence changes that are consistently found in affected individuals but are absent in unaffected individuals. All right? That's the basic proof. Now, there are lots of other things you need to do once you find such a gene. You have to do all sorts of things. You do gene expression. I should tell you I should also mention, why do you want the gene? Well, because you can use the gene to find out all these other things. You know the gene is the cause of the disorder. A variant form of this gene is the cause of this disorder. But that may or may not be that enlightening. A large number of these genes that were identified by this positional cloning In the early days, they didn't look like anything anybody had ever seen before. It was not obvious how this gene caused this disease just by looking at the gene itself, all right? So what do people do? Well, they look at gene expression. Where is the gene expressed? So for instance, with stuttering, there's a question, is this a disorder mostly of the brain or of a disorder of the musculature of the speech-producing muscles or the nerves that innervate the speech-producing muscles? Well, you can look to see if we find a stuttering gene, whether it's expressed mostly in the brain or mostly in the muscles or mostly in the peripheral nerves that connect the brain to the muscles, all right? And so you can begin to answer these kinds of questions with gene expression studies, all right? Gene function, what does the gene code for? What does this thing look like? Does it look like a structural protein, something like cartilage that makes, a structure in the body? Does it look like an enzyme, something that runs chemical reactions in the body? Okay. What is the biochemistry, the physiology, that is the role of this normal gene in the function of our body? What is the pathology? What happens in the body when something goes wrong with this gene? All right. And finally, my goal, coming from many years in the private sector, is to think about the development of therapies. Because when you have the ultimate cause of a disorder in your hands, you can think about how this could be exploited to develop things to correct the problem. All right? All right. So why is positional cloning a good approach for speech and communication disorders? Well, speech and communication disorders are highly complex, uniquely human functions. They're highly dependent on activity within the brain, which is a very difficult organ to study in live, intact humans. No reliable animal models exist for these disorders, and other approaches have seen limited success in identifying the causes of communication disorders. So I don't, you won't find a bigger booster of genetics than me. But I do, in the interest of full disclosure, I do need to tell you that, of course, the situation isn't perfect. There are some drawbacks. There are some problems with using genetics. The first is speech and communication disorders typically do not show simple inheritance patterns. That makes identification of co-inheritance much more difficult. In addition, it's probable, it's in fact, I think, likely, that any one DNA sequence change may not be necessary and sufficient to cause the disorder. These things probably aren't going to be like hemophilia or neurofibromatosis, where if you have the mutation in the gene, there is a 100% chance that you will then have the disease, okay? And you can't get the disease without a mutation in that gene, all right? They're probably a little muddier. There are probably more than one gene. Some people are, in fact, there are probably a number of people walking around or in this room tonight who have such a gene for a speech and communication disorder, but manage to recover spontaneously in childhood. Or, in fact, might not have even ever expressed a communication disorder, but they're still carrying a gene, all right? That's probably why the inheritance patterns of these things look a little bit muddy, all right? Okay, so I want to tell you now, though, about some examples that that will hopefully make you as excited as I am about the prospects of using genetics to find actual genes for speech and language disorders. The first one is very famous and has to do with this family, the KE family. The KE family was identified in Britain some time ago, a couple of decades ago. This family has an unusual speech disorder that's transmitted as a simple genetic trait. For those of you who are taking biology, it's inherited as a simple dominant trait in this family, all right? So half of all the people in the SIB ships get it, and it never skips generations. It's always, if someone has it, one of their parents also always has it, all right? All right. What is this disorder? There was a lot of dispute about what exactly this disorder was for a long time. These people have a complex disorder. They have both a dyspraxia, they have an inability to properly produce speech sounds and to link these sounds together into clear, intelligible words. All right? And they, in addition, have another problem. They clearly have problems organizing words to make clear sentences. The standard rules of grammar that we all follow, subject, verb, object, these people have problems with. However, they are of normal intelligence, all right? This is not a mental retardation problem or some more global deficit. These people are more or less, other than this speech problem, more or less normal. And they don't seem to have anything else wrong with them. It's not what we call syndromic. It's not this plus something else, all right? So a linkage study, a genetic linkage study was performed, and they did markers up and down all the chromosomes, and lo and behold, they found linkage with markers on human chromosome 7, all right? And in fact, that allowed them to find the gene. That gene is now called the speech 1 gene, and what does it do? Well, it's actually quite a remarkable story. It codes for what geneticists or biomedical scientists would call a transcription factor. It's something that turns genes on and off in your DNA. It controls which genes are expressed where, all right? So the speech 1 gene encodes for essentially a genetic switch, which turns on a specific set of genes during development of the brain. And so now there's many exciting things happen. I don't know whether you saw it. It was picked up by the newswires a couple of weeks ago. They have studied variation in this gene and shown that the common version of this gene, that humans have a unique set of alterations in this gene compared to all the primates, compared to all our recent evolutionary ancestors. And they were able to date the approximate time of the acquisition of these alterations, and these authors postulated that the acquisition of the alterations in this gene was what helped give rise to speech as a uniquely human trait that separates us from our nearby primate ancestors. In any case, it's a great story, and for instance, it will be important to see exactly what parts of the brain this is expressed in, because it's going to tell us what parts of the brain are specifically important for aspects of speech at the very micro level that's otherwise very hard to get at, all right? Okay, so I mentioned stuttering, which is something I work on. I'm going to tell you a little bit about stuttering. So stuttering, as I mentioned, has muddy inheritance. It's clearly genetic, but it's not always muddy. There are a few cases that show clear, apparent single gene inheritance. The first is the so-called family X. It was discovered in Iowa in the 1940s, and it looked like a simple single gene trait. There's a family that's called the Utah family. Many of them actually live in Idaho. They're very well described because they have a large, well-known pedigree. Lots of brothers and sisters for many generations back in this family. And this family has actually probably a unique disorder that's stuttering and a mixture of other things. But it's clearly inherited as a relatively simple genetic trait. And then finally, a family in equatorial Africa, which we've found quite recently in Cameroon, which clearly has simple, plain, non-syndromic stuttering inherited as a very simple, what we would call dominant trait. These results suggest that at least some stuttering is caused by simple inherited factors, although probably not all. What sorts of linkage results do we have for stuttering? Well, there are some results out there. There are some results that have just come out, and there are some that are in press, and there are some studies that are in progress. And they are as follows. There's a linkage study that was done in a very special population, the South Dakota Hutterites. They were a very religiously isolated group. The whole population was founded by fewer than 100 people, and they have only married within that group for the past 7 generations. There's a fair amount of stuttering, and they did a linkage study and found 3 locations, one on chromosome 1, one on chromosome 13, and one on chromosome 16, that seem to carry the gene or genes responsible for stuttering in this population. We, in our laboratory, did a stuttering linkage study in the general North American population and came up with a location identified on chromosome 18. We have clear evidence that it's at work in one large family and may be at work in many other families, but that at the moment is still under investigation. I should also tell you that us human geneticists frequently go to unusual places in the world where family structures are different than they are in the United States, because those can be very powerful for doing a linkage study. So we have a set of 70 families that are the results of either first or second cousin marriages, where there are many individuals who stutter in each of these families. And we're doing a linkage study in those from Pakistan now. And we're working on this large family from Cameroon now. So it's clear that people are getting linkage results in stuttering. It's clear that we've got more places we can look where we have a reasonable expectation of getting more additional linkage results. So I think we're on at least the first step to finding genes that can predispose individuals to stuttering. Okay, I also want to talk about specific language impairment. SLI, as it's known, is defined as the failure of normal language development in the absence of any other deficits. All right? It's common in young children. As many as 5% of people display SLI in the schools now. It's associated, has been associated with what are called auditory processing disorders. Auditory processing is roughly defined as what happens to sound after it reaches your ear. Okay? Because your brain takes the sound from the ears and actually processes it to produce what we perceive as sounds. All right? And this is probably associated with SLI. In addition, a large number of these people will develop characteristics of dyslexia. Specific language impairment has had some linkage studies. There's one recently published in England, 473 people from 98 families, a very large study. And they demonstrated linkage to chromosomes 16 and 19. It's not clear whether this 16 is different from the gene in the Hutterites on chromosome 16 that causes stuttering. We don't know yet. It's possible it could be the same gene. In addition, a special group of authors in Canada, 86 individuals in 5 families of Celtic, largely Irish origin that lived in eastern Canada and demonstrated linkage on chromosome 13. Whether this is the same position as the location on chromosome 13 that causes stuttering in the Hutterites also remains to be seen. Okay, and finally I want to talk a little bit about disorders of auditory processing. That's what happens to sound after it's received by the ear. It includes pitch, or tone recognition, and temporal timing processing. The reason we're interested in auditory processing is because it's required for the understanding of speech sounds. And people with severe problems in auditory processing tend to have speech problems, all right? So I'm going to talk mostly about pitch recognition. Pitch, do, re, mi, is a remarkable human ability. It's very fast. It's very sensitive. It's actually one of the most powerful in terms of its resolution power, how small a difference you can perceive. It's the most powerful of our human senses, all right? Now, a perceived pitch is the result of sound processing by the listener. What we hear is what happens is that sound comes through the air, reaches our ears, and then it is processed to produce what we hear as a pitch that's actually perceived, okay? So there's a lot of processing that goes on in order for us to tell do from re from mi, all right? Humans vary widely in their pitch perception abilities, from absolute pitch, sometimes called perfect pitch, to what we sometimes call tone deaf. And so the question is any of this variation due to genetics, due to inheritance? So we developed something called the distorted tunes test. It's a measure of the ability of the listener to detect mistakes in simple popular melodies. It's a short test. It's 26 melodies long. We put it on a compact disc, and people are asked whether the melody you hear is correct or whether it has mistakes in it. It's real simple. 26 questions, either yes or no, all right? And we also ask them whether they're familiar in case they don't come from a Western musical tradition, so they don't really have a very good sense of Western musical rules, all right? So I'm going to give everybody some examples here. First thing I'm going to give you is an example of a correct melody, all right? This one is Amazing Grace. Okay, so I trust that most of the people here in the audience could recognize that there's really nothing amiss with that particular melody. I'd like you to contrast it, however, with this, which I'm not going to tell you the name of, but I predict most of you will be able to tell what it is. So that was Clementine, with some fairly substantial mistakes in it. And in fact, most people can easily recognize that something is very seriously wrong with this tune. Now I'm going to play a couple of others, and basically we can do this as a little test. I'm going to play a pair of them and have everybody listen, and then you can decide for yourself what you think. Okay, so I trust that was familiar to everyone. Now, I'm going to give you another one as it actually exists on the Distorted Tunes test. So, I trust it's pretty obvious that the first one was America the Beautiful, played correctly, and the second one was America the Beautiful, played very incorrectly. And most of us can tell this, but remarkably enough, they actually don't sound incorrect to everybody. I should tell you that some nice things are known about the Distorted Tunes test. For instance, it's known that scores change little over an individual's lifetime. Your scores don't change on this test. You can practice and get a little bit better, but you don't get much better. And the performance of males and females is the same on this test. So how do people do? Well, this is the performance of a whole bunch of people, and this is the score. So this is 26 correct. This is a perfect score on the test. And you just pay attention to the red lines here, because that's the combination of everybody. And as you can see, most people do pretty darn well here. But random guessing, so there's 26 questions. If you just threw darts at the answer sheet, you'd get 13 right. Okay? So down here at random guessing, there are actually a fair number of people down here. As a matter of fact, about 4% of the population is unable to distinguish correct from incorrect. They score no better than random on this test. So we use this as a twin study. We did a lot of people. We did 136 pairs of identical twins and 148 pairs of fraternal twins. This twin study originally started as an osteoporosis study, so that's why they're all female, although we've shown that there's no difference between males and females in this test, and we take females as a pretty good representative of the whole population. We administered the Distorted Tunes test in a little test called the 5-minute hearing test, which is not a hearing test. It's a questionnaire designed to identify people who have serious problems with their hearing that might otherwise impact their performance on a test like this. All right, so what are the results? Well, first of all, the performance on the Distorted Tunes test is largely due to your genes. So there's something called the heritability, which is basically can be thought of as the fraction of it that's due to genetics, is between 70 and 80%, depending on how you classify people. This is a very high heritability. To give you some idea, the heritability of your cholesterol level is heritable. The heritability of your cholesterol level is about 30%. In fact, the only thing that is reproducibly higher heritability than this in humans is how tall you are, which has a heritability of about 90%. Okay? But all sorts of things that clearly run in families, diabetes, obesity, asthma, have heritabilities in the range of 0.5, well under this. So it's extremely genetic. Your performance on this, your pitch recognition is very genetic. Shared environment contributes very little. The correlation between Distorted Tunes test and the 5-minute hearing test is very poor. It's about 0. And that means that musical pitch recognition has little to do with your hearing, as hearing is traditionally measured. All right? So what's the next step? Well, twin studies estimate how much of a trait is due to genetic factors. But it does not answer lots of other questions. It doesn't answer what's the likely mode of inheritance. How is this thing inherited? It doesn't answer the question whether there's a major gene at work. It doesn't answer the question of how many different genes could contribute to this trait. All of those, to answer all of those, you have to study families of affected individuals. So we've gone ahead and found some families. We've done this at Brigham Young University, which is a parochial school. It's run by the Church of Jesus Christ of the Latter-day Saints, which is popularly known as the Mormon Church. Mormons tend to have large families, well-documented family history, family structure, and everybody who goes to BYU, to Brigham Young University, attends devotional at 11 a.m. every morning, and they all sing. So they have universal exposure to music. If someone does poorly on this test, it's not because they haven't heard these things many times before. We administered the distorted tunes test to 180 random volunteers and found, with what we expected, about 40 individuals with very poor DTT scores. So then we went and looked at their family members, all right? So now these are just family members, and we asked ourselves, well, how do they do on this test? And here are some examples. Those of you who aren't used to looking at pedigrees, the girls are circles, the boys are squares. This is dad, this is mom. They have 6 kids. The oldest is a daughter, the youngest is a son. So here's one of our guys that we found at BYU. He scored a 15. His older sister, who we had no connection with, scored an 11. All right, basically random guessing. And then 2 other sisters who didn't do so bad. Here's another family where we found a guy who scored an 18. Well, his parents are both fine. They did almost perfect on the test. But he's got a younger brother who, in fact, did quite abysmally, and another brother, the oldest brother, who isn't much better, and then these guys who might be kind of intermediate. All right? So the answer is you can find families in which deficits, tone deafness, if you will, behaves like a simple genetic trait. And it will be possible, we believe, to do genetic linkage studies and ultimately, we hope, to find the genes responsible for this disorder in these families. All right? So what are my conclusions? Well, 3 take-home messages. Number 1, genetic factors make a significant contribution to human communication disorders. 2, today's genetic methods and technologies are allowing us to find these genes and to learn what they do. 3, understanding the causes of these disorders, that is, the genes that cause them, is an important step toward improved treatments for these disorders. So with that, I will stop. Thank you very much for your attention, and take questions.
With the identical twin studies, were all the twins stutterers? You didn't mention that.
Let's see.
You said they were 2 to 3 times more frequently both stutterers compared
Correct.
to
So are you asking whether all identical twins both stuttered?
Yes.
So the way geneticists would say that is the concordance with identical twins 100%? And the answer is no. It's not 100%. The identical twin concordance is high. It's over 80%, but it's not 100%.
I just have a basic genetics question. In identifying the human genome and all the individual genes, are there punctuation marks so you know where one gene ends and the next begins, because you have the 4 amino acids? And
Right. So the question is, are there punctuation marks in the human genome sequence so you know actually what are the genes? Actually, that's quite a sophisticated question. It turns out most of the DNA in our chromosomes doesn't code for genes. Perhaps as little as 5% actually actively is genes. The rest used to be called junk DNA. There are those of us who don't believe it's junk. There are those of us who believe that lots of it is actually just things we haven't figured out what it does yet. But it's clear some of it is not genes.
Right.
Did you have a question?
Yeah, I did. I was looking through the papers that you guys handed out, and it mentioned the Turner syndrome. When females are XX-Y, I believe. Is that it?
X blank.
I think so, yes.
And it said that
Is it XO?
X blank.
XO,
X-
right.
X-
It's an
So
XO,
X
right?
Is X and nothing else. They just have one X.
Klinefelter's.
Right, thank you. Klinefelter.
All right, so it's XO. Does that show any, and does it indicate that it has something to do with, sex linkage, not just linkage groups?
Right, so sex linkage, so yes, all of the genes in that case are sex linked.
Right.
So sex linkage was the first kind of linkage discovered. And sex linkage was uniquely recognized early on because the genes, there are 2 special chromosomes that we all carry around called the sex chromosomes. One's called X and it's pretty big, and the other's called Y and it's really small. And the X boys have an X and a Y, and girls have 2 X's. And people who just have 1 X and no Y have a disorder and called Turner syndrome. And part of the manifestations of this disorder are probably due to the fact that they don't have the right dosage of the genes that are on the X chromosome. So these sorts of disorders that occur only in one sex, are sometimes known as sex-linked disorders, and it was the first linkage observed. But the short answer is yes, things that are on the sex chromosome do have a unique pattern of linkage, and they display this sort of behavior.
Okay,
Thanks.
Think forward. Think Research Channel.